Good morning, and welcome to the Land Grant Seminar Series 2024 Spring Semester. And we're on week 13, right, Dr. Owl, or 12? 13, week 13 of the semester. And the semester is winding down, so we have two more seminars to go. In the beginning of this seminar, I would like to thank our researchers, students, and faculty, and our administrators for supporting the Artificial Intelligence in Agriculture Conference tour, and also the conference itself. We had a very successful tour here. Our scientists shared their information, and the student as well, in a poster session across the hall from here. Also, we toured the MEETS lab, we toured the National Goat Center. We ran out of time. We couldn't see other facilities because they came late and they have to go back to reach there because the program starts at 5. So overall, we had a great time. And Vice President for Research, Magesh Rajan, he was here. He supported us and his staff. So we were very thankful, and our student performed very well. At the same token, we at the conference in College Stations, 6 of our students and in fact 10 faculties and students, undergraduate, presented their work there and interacted with the participants. We received lots of good comments. People were commenting about our student performance. Malani, Obimon, and Katana, and others. We had a very good. They were impressed with the level of presentation and quality of their presentation as well. So I extend my gratitude to our dean and our associate deans for their support. They supported the conference at the platinum level, no less than that. Thank you. In fact, we had 329 participants. We had this, I think the statistics are here, but we had a 90 presentation and 76 posters. So it was a very good conference, and I really pleased with everything. So thank you again. And it's a good model of working with College Station and organizing a great event like that at the national level. So it was a great opportunity as well. So hopefully we can build on that for future events. Next year. The conference will be next year. In fact, there is no nationally here, no AI conference by itself as a stance. And AG is taking the lead. That's not surprising. AG always have been in the lead. So yes. And Vice President Rajan mentioned that. They said we need an initiative at the campus center, and AG is leading the way. So I think that speaks for the depth of and support that we get from our administrators in this regard. So tomorrow and the next day, we will have the field day, the AG field day. So please make sure to participate there and be active and connect with our presenters and our stakeholders. As I said, we will have a couple of more presentations. One is from Dr. Svitcha. He's a very well-known professor of physics here on campus. He's an excellent one known internationally. And the last seminar will be with the director of USDA Southern Plain Climate Hub. He will be coming on May 2nd. That's another great opportunity to connect on another important topic, which is climate change and climate smart agriculture, etc. So that will make the full for this spring semester. I'd like to introduce our speaker for today, Dr. Danisha Parker. She received her bachelor's degree in biology from Rust College in Holly Springs, Mississippi. During that time, she participated in many research internships and became a McNair Scholar. She gained a passion for student research and took advantage of the opportunities it was offered to her. She went on to go to get a PhD in entomology from The Ohio State University in 2021 and joined Prairie View A&M University in 2023. Her research includes studying habitat change in urban ecosystems. Dr. Parker serves as the Student Research Development Officer. She's overseeing the research and innovation of a Scholarly Excellence Award program, RISE. To date, while under Parker's supervision, over 120 undergraduate and graduate students have had the chance to participate in research across campus. With that, I would like to welcome our speaker. She will be talking about drivers of predatory insect distributions with a legacy city. Dr. Parker.
Thank you. Thank you. Good morning. Dr. Parker. Just to give a disclaimer, my research, I identify as an entomologist and an urban ecologist. So my research is basically both of those put together. So a little bit on the social aspect, but definitely agriculture and science as well. Just to make sure, which one would be?
The one on the right, I think.
Okay, I got it. So today I'm talking about drivers of predatory insect distributions within a legacy city. So just to give a background, urbanization is increasing worldwide with over about 50% of the population residing in cities. This becomes an issue for many of the species that live within those cities. Due to transformation to build surfaces, many species are unable to cope with the changes within their habitat. So their habitats are reduced in quality and quantity. Not only that, they're also facing competition from many exotic competitors that are introduced into an urban landscape once you build it up. So they're fighting to, native species are fighting to excel or strive in these cities. And then they also have many issues. This is going to decrease the biodiversity throughout the city and kind of create biotic commoditization, which is basically when there's one top dog, right? One dominant plant species, one dominant animal species, one dominant insect species. This success of exotic species is predicted to be a part of the disturbances that these native species are unable to withstand. But despite these documented challenges of life in cities, cities are recognized as undervalued resource conservation efforts. Many cities are finding new species that have never been found before all across the world. So the needs to kind of protect these habitats in these cities are very important. This is going to be particularly true for post-industrial cities. Post-industrial cities is any city that was once really dominant, but due to the fall of some industry, they have a population decline, which leads to overabundance of vacant land within these cities. One such city is Cleveland, Ohio. Cleveland, Ohio is about an hour and a half from Ohio State University. This is where I did all of my research. In about the 1920s and 1930s, Cleveland, Ohio was booming. They had the automobile industry, many jobs, people from all over the world were coming to Cleveland, Ohio. Then there was an automobile drop, right? All of the industries moved overseas. That created a loss of jobs for many people in the community. With no jobs, people are leaving. There's no need to stay. In about 10 years span, Cleveland's population dropped in half. That leads to an abundance of vacant land. We'll talk a little bit about that vacant land. But to date, because of this drop in population, Cleveland, Ohio has over 27,000 vacant lots throughout the city. These, the red represent all of the vacant lots within Cleveland, Ohio. This is a lot of green space. This number increased annually by 1,000. I graduated from Ohio State in 2021. So if this number was 27,000, it's about 30,000 vacant lots within Cleveland, Ohio. That's pretty unique for a city, but how does the vacant lots get there? So basically, if everyone is moving, there's no jobs, there's no need to stay in Cleveland, right? You move out of Cleveland, now you have an abandoned house. The likelihood of someone moving into Cleveland and buying your house is slim to none because there's no jobs. So what the city does is demolish the house. This create vacant land throughout the city. This is what the 30,000 vacant lots around Cleveland look like. They are simply mowed once a month. That's really costly to the city, and that's a lot of work to mow 30,000 vacant lots once a month.
We can send some goats.
I kid you not, I saw a goat in one of the vacant lots in Cleveland, Ohio. I have a picture and I'm gonna find it and I'm gonna send it to you guys. It's crazy the things I saw there. So my lab, we took advantage of this opportunity to re-image some of these spaces into conservation resources. So with $1, we rented 64 vacant lots from the city of Cleveland, Ohio for 8 years to do research. We transitioned these vacant lots to pocket prairies, which are basically patches of native grasses and forbs that we feel can be beneficial to the city, community gardens or farms that can provide produce to the community. We also changed them into some rain gardens that can collect some rain runoff, but also be used as a green space for the people in the city. And as an entomologist, right, we wanted to see, okay, the urban ecology side, but how is this really helping the biodiversity of the insects that's within Cleveland, Ohio? So I wanted to see what factors was driving the insect distributions. And I had 3 ideas in mind when I thought of my project. Landscape, habitat management, and the interactions that these insects are going to have with other species within there. So today, I'm going to kind of break down the 3 chapters of my dissertation on what I looked at. So I really looked at native lady beetle decline, how this was related to exotic species invasion in these areas. I also wanted to know what ecosystem services that these insects are providing in these habitats, as well as some conservation efforts that we can implement to help the city with the problem of these 30,000 vacant lots that they need something to do with. And then we'll talk about at the end what I found and was it helpful, was it beneficial? So I'm going to walk you through each chapter. This is kind of the basis of my research, this model in general, so I'll walk you through that first. My first question was, can these urban habitats even support declining species? If that's something we're worried about, do we even have the opportunity to do that somewhere here? Are the declining species even there at this point? And I had a little idea, right? So the urban species pool in Cleveland, Ohio, so I'm talking about any insect species within Cleveland, Ohio, is going to be filtered by the landscape, the way in which we manage the habitat, and the interactions that it have with other species within that habitat, right? So that's basically the filtering process to what you may find in this vacant lot compared to that vacant lot. So keep that in mind throughout the entire presentation. It must have been good. That was published in Urban Ecosystems in 2020. And I could just give you a brief background of what I find, and you guys can go read it as well. So one key species was lady beetles. If you're in agriculture, if you work, you know lady beetles are important agriculture predators. They're important predators in general. They kind of help us control any pests that we find within these settings. But worldwide, native lady beetles have experienced a dramatic population decline. This has been documented for years and years. To date, both the 9-spotted and 2-spotted lady beetle that was like exceedingly popular are now rare. You can barely find them, especially in Cleveland. And many of these declines coincides with the establishment of exotic species that were brought over as biological control agents for agriculture. To date, across the US, we have 4 exotic species that dominate the total lady beetle community. And these changes are now sometimes due to intricate predation, but we also think that there's other things that make exotic species thrive more in these areas. So I had 2 hypotheses when looking at native and exotic lady beetles in Cleveland. My first, and this was me thinking that landscape variables would be weak filters, right? Just because the research that was done on lady beetles. So I thought that the habitat and the interactions that the lady beetles were having with exotic lady beetles may influence their decline. So I'll give you an idea of my study design. So now we're using these pocket prairies. I have 4 different treatments for this particular study. The first being a control treatment. I basically managed the vacant lot the same way the city did, mowed it once per month. The second is a successional treatment, which is basically letting whatever weedy vegetation that exists in that vacant lot grow throughout the growing season. We're not touching it. The third is a low diversity prairie. So we have 3 native grasses and 4 native forbs. And then the next is a high diversity prairie with 3 native grasses and 16 native forbs. So a little more diversity in that high diversity one.
So these are native to the
Ohio,
to Ohio.
Cleveland, yes. So within these vacant lots, I established a 7 by 15 meter quadrant. I put 5 sticky traps out during the month of June through August of 2016 and 2017. I had a total of 32 sites that I was working with each month. Those 5 lady beetle traps were left out. I collected and identified the lady beetles as well as the prey. And for prey, I use aphis because that's the known prey of lady beetles. We also took a look at some vegetation variables within each lot. We estimated the biomass that was in the lot. We calculated the average bloom abundance that was present within the lot and also the average plant height. We also looked at some landscape variables using GIS. We calculated the percent of pavement surrounding the lot and how far one green space was to another, so patch isolation. My statistical analysis was carried out using a generalized linear mix model just to look at the difference across treatments, but I used a PSLCA analysis, which is going to allow for me to have multiple response variables, a large number of predictive variables that may be collinear, as well as a small sample size relative to the number of predictive variables, because I was dealing with a lot of predictive variables for this particular study. And I was the one actually out there doing the work. That's me. I guess I'm doing plant height here, looking at the average plant height. And we found some cool things. First, we found 12 species of native lady beetles, representing only 28% of the total lady beetle community across all sites. We also found 4 species of exotic lady beetles that we expected to find, but they dominated the lady beetle community. The most common species was the native orange spotted lady beetle, which is here, and the exotic 14 spotted, and I'm giving you guys the general names for easier context, and the 14 spotted lady beetle here. We found absolutely no difference of lady beetle abundance across treatments, but it's okay. This was our first year establishing those prairies. That was kind of almost expected. So now I'm going to walk you through the results of my PLSCA. And this is when I'm looking at my vegetation and my landscape variables. So I'm going to start off slow. So these are my response variables. So I'm looking at how those variables affect the abundance of both exotic and native lady beetles. And I'm looking at it separately because I want to know, are they having those same drivers that is affecting the abundance? I'm going to walk you through. The dotted lines represent a negative correlation. The thicker the line, the more negative that correlation is, or the more strong that correlation is. So first, I looked at impervious surface around each site. Impervious surface is simply the amount of pavement that is surrounding each site at a 500-meter scale. We saw that this negatively correlated with both exotic and lady beetle abundance. So lady beetle abundance decreased in sites that were surrounded by impervious surface or pavement. We also looked at patch isolation. Patch isolation is saying how far one habitat is to another green space habitat, right? So how much impervious surface is in between. And we saw a decrease as patch isolation increased. There was a decrease in native lady beetle abundance and exotic at a 500-meter scale for both, but at a 1,500-meter scale, it still affected native lady beetle abundance. So even as far out as 1,500 meters. And I use these to account for lady beetles' dispersal ability. So when going back to my hypothesis, I predicted that landscape would act as weak variables of lady beetle distribution. That was not the case, as we saw. And I also thought that local scale variables such as vegetation height, bloom abundance, and availability of prey will affect lady beetle abundance. But when doing my PLSCA analysis, none of those mattered. So in this idea, if I'm thinking about hierarchical filters, landscape was the one that was driving Lady Beetle, the local community that we found within each site. And local scale variables were not strong filters. That was a little disappointing, right? Because we spent a lot of money. We say we spent $1 to buy the lots, but the man labor to maintain 64 lots is a lot. So we're like, okay, but you have to think we're in a lab where multiple people are doing different projects, different insects. So I said, okay, maybe the prairies aren't a big deal. Let's go another angle. And the angle I decided to go was urban agriculture. So the question that I wanted to ask was, does the transformation of these vacant lots to urban farms result in the recruitment of beneficial insects? We need beneficial insects to have a vital or a viable farm, urban farm. So food insecurity affects 49 million people. Access to nutrition is very limited in inner cities. When in Cleveland, Ohio, I don't think I've ever saw a grocery store, but I saw a convenience store and a McDonald's on every corner. So them having access to local nutrition was a huge issue. But the thing is, natural enemies face stressors within cities. They have pollution, they have habitat isolation, things that you just don't find in rural agriculture. So they have new unique problems that we had to address. So the first thing first is, do they even have the insects or the systems to have a good urban farm? Lady beetles are beneficial insects in urban farms. They feed on aphids, thrips, and spider mites that are all pests to many of the crops. Exotic lady beetles have been introduced in the US for biological control agents for years. So we know that they're important in biological control in farms. So I wanted to know if there was a difference in the recruitment of lady beetles on an urban farm compared to that in our vacant lots. So I hypothesized that lady beetle abundance will decline in urban farms relative for vacant lots. But I thought this because of just simply the disturbance that happen in farms. There's a lot of disturbance. Lady beetles typically are either on the ground or on plants. So I'm thinking that maybe it's too much disturbance for those lady beetles to actually be of any use in these farms. I was ready this time because my previous research showed that landscape was the key factor in lady beetle abundance. So I hypothesized that even within farms, there will be a decrease in the lady beetles that we find due to increased impervious surface that surround each site. But I added one more thing when dealing in farms, and that's contamination. Cleveland, Ohio is a legacy city. It had many factories that was pollutants. If we're saying those factories have been torn down and demolished, that debris is generally left in the soil, the same soil in which we put an urban farm for the community. So that's a big issue. So we also want to look at contamination. And I hypothesized that lady beetles would decrease in habitats that are more contaminated than others. And I did something a little different with this study because I had to say, if I'm saying I want to look at farms, I have to see what's happening in the rural farms that surround in Cleveland. So with this one, I looked at the vacant lots that was mown monthly, the urban farms, but I also studied some rural farms that surrounded the city, as you can see here with the purple boxes. So I had 21 sites in total for this study. This was done during August 2018 and 2019. Once again, I established my 7 by 15 meter plot in the center of the lot. I placed my 5 sticky traps out for a week. I did my vegetation. I looked at my landscape variables and my contamination variables, and we used the pollution load index to kind of calculate the contamination, and just basically using a soil rod and then sending it off to the lab, and they let us know how much pollution is present within each of these lots. And once again, I use a generalist model and a PLSCA because that's just the holy grail for this particular research project. And I found almost something similar. So I found 7 species of native lady beetles, but they represented over 40% of the lady beetle community within these years. Once again, the 14 spotted lady beetle was the most dominant. But the orange spotted lady beetle had another native lady beetle that came to play, and that was the pink lady beetle, which is so pretty. And that made sense because I know for a fact that this pink lady beetle feed on mostly aphids. So of course I'll find them in rural farms and urban farms more than the orange spotted lady beetle. Once again, there was a similar overall abundance across treatments. But we still want to look at the PLSCA and see how those local and landscape variables affected lady beetle abundance. And in this case, I didn't separate native and exotic lady beetles because when you're dealing with an urban farm, it really don't matter, right? May the best win in that case, the best predator. So I looked at not only the species abundance, I also look at the richness, so how many different types of lady beetle species we found within these lots. And I also want to focus on only those lady beetles that I know feed on aphids because I know they are predators that are going to be beneficial for these gardens.
Mhm.
And once again, we're doing the same thing. The dotted line represents a negative correlation. The thicker that line, the greater that correlation is. Patch isolation was the key driver for all three. So as the patches, as more pavement increased around a patch, we saw, patch isolation, as patches were farther from other green spaces than others, we saw a decrease in lady beetle abundance, lady beetle richness, as well as aphid feeding lady beetles. Management did not result, it had nothing to do with the recruitment of lady beetles into vacant farms. Landscape was a big driver in this, and fragmentation was just a problem in terms of lady beetles. Another surprising thing is that contamination also did not prove to be a strong driver with lady beetle abundance. And this caused me to think a lot. And what I thought was a lot of soil is reintroduced when you're establishing a farm on land. A lot of them have raised beds or different things going on. So just that introduction and turnover of soil could be a reason why the pollution didn't serve as a big issue with the their abundance or richness. I wasn't done. So at this point, this is my last one. You guys, at this point, I wasn't done. 2 studies have showed that landscape is a big driver with lady beetle abundance. But lady beetles aren't the only beneficial insect predators in Cleveland, Ohio, right? We have a lot of beneficial predators. So another one, I want to look at something else, right? Let's see if this is applying to other insects as well. So this title was, Does Urban Landscape Fragmentation and Habitat Transformation Influence Dolichopotidae Abundance and Dietary Niche Breath? Dolichopotidae, I'm going to call it a long-legged fly, is this pretty fly. It really looks like this, you guys. That's a good picture of a long-legged fly. And I looked at the dietary niche breath of the Dolichopotidae. So I kind of added a molecular component to this particular chapter. Food is a key opponent for insect health, just like it's a key opponent for our health. It is considered one of the most important dimensions of dietary breath, and what we deem as important. Dietary niche breath, is a measure of basically the diversity of the dietary resources or sources that an insect will feed on. So an example would be like an insect that has a specialized diet means that they only feed on one thing. An insect that has a generalist diet will feed on multiple things. This will be an increase in dietary niche breath, right? Because what they feed on is a wider range. In cities, we know that both habitat and landscape can influence prey that's available to beneficial insects. It might also restrict the realized dietary niche breath. So maybe they're feeding on only what's available and not necessarily what they would feed on if they were in a different setting that wasn't of season, in the area. I use Dolichopotidae because they are also a common predator group that are found in natural habitats, agroecosystems, and urban green spaces. They have a really generalist feeding habit. Dietary niche breath has been analyzed on Dolichopotidae in agriculture settings, never in a urban setting. And I use the Condylysis long-legged fly just because it was the most abundant within our habitats within Cleveland. So my hypothesis was that first, because I knew that Dolichopotidae had a wide, or the long-legged flies had a wide dietary niche breath, I assumed that we will find something very similar in urban systems. I also predicted that the transformation of these habitats, as well as the surrounding landscape, will have an influence on what's available or what we find in their diet. So this was a little more difficult to collect. Because I'm doing molecular work and I'm risking contamination, you guys, I had to hand collect flies. How do you hand collect a fly? So I'm literally like using an aspirator and trying to like suck the aspirator through a straw that's filtered so it can't go in my mouth, and then dump it into a jar, close it, put it on ice so that it's not degrading before I could get to the lab and get it into the freezer and actually use it. So it was really difficult collecting flies. I did this through 10 a.m. to 2 p.m. So in the beaming sun in the middle of the summer, collecting live flies for molecular work. Luckily, the abundance was done using pan traps, and I was just able to sit those pan traps out during that time, and within 4 hours, the pan traps will be filled with flies. And I used that to collect for abundance. I also took a look at the same landscape and local variables. I collected only 10 individuals per site, which was still a lot, a lot to do. And I used the standardized Levens measure to measure the dietary breath that was present within each of those lots. Cool results, you guys. Treatment mattered, okay? So the first thing when doing my generalized linear mixed model, I found that long-legged flies were actually more abundant in vacant lots compared to prairies. That was really surprising. So the vacant lots were winning, but I also got some cool results. And just a disclaimer, so how the me getting the dietary niche breath worked was I basically completed the DNA extractions. Ohio State has a sequencing center. After I do the extractions and prep them for sequencing, they basically sequenced it. They give you back some results you don't understand, but luckily I had a collaborator, Rodney Richards, that helped me kind of barcode, and I had some meta barcoding experience, so we kind of worked together for this particular project. And this is basically what we found in terms of dietary niche breath. Each box represent like a different species that were found within the the flies, and this is all. But there was only 3 groups. And by groups, I mean the type of insect that we found within the flies. The purple color represents Diptera. Diptera is basically saying other flies, right? They're flies. So this is how many different flies we found within the guts of these long-legged flies. We found some true bugs. True bugs will be something like an aphid. We found that, and this is just other stuff that was found in a small amount, not enough to kind of pull out. So we found that out of all the Dolichopolidae, although it's not a super broad dietary niche breath, they're not eating the same exact thing. Something cool that we found was that one of the most abundant things that these flies ate was mosquitoes. And if you know anything about mosquitoes and urban systems, that's the whole research project in itself. So that was really interesting to find. I also did the PLSCA, and once again, I did the dietary niche breath, so how broad that diet was, as well as just was there even prey found within that particular fly. So the percent of prey that was positive. So for this one, once again, the dotted lines represent a negative correlation, but the solid lines represent a positive correlation. The thicker the line, the greater that correlation. Landscape drivers was a negative correlation. As the pest isolation and the amount of pavement increased around the site, we saw a decrease in both the dietary niche breath as well as the percent of prey that was found positive in the guts of these animals or these insects. But some local variables start pulling out. So we saw that plant height, biomass, and bloom was important vegetation variables. So for an example here with the yellow. So as the average number of blooms increased in a site, we saw a decrease in the dietary niche breath. That makes sense because we saw them more abundant in vacant lots compared to the prairies that would have had greater blooms. Then for plant height, it was a negative correlation for dietary breath, but at least they ate something, right, in the prairies. So they were still eating the prairies. They just wasn't eating a lot of different things. So they probably had a more specialized diet, which is why they preferred the vacant lots, because they had a greater abundance of food to eat within those sites. So this was some really cool results. And the same thing happened for biomass, right? We're expecting to have more biomass in the prairie compared to a vacant lot. So it makes sense that we found a greater, a decreased dietary niche breadth in those areas. Dolichopotidae didn't have a broad dietary niche breadth, only eating 3 groups of insects. That's not a lot. We have multiple groups. So that wasn't a broad dietary niche breadth. But local habitat quality as well as habitat fragmentation did influence their dietary niche breadth, which has showed us that these farms are important or these transformations to these green spaces are important. Different things are going to be found when looking at different taxa. Something cool that we wasn't expecting was that our findings highlight the value of long-legged flies as a mosquito predator. That was something that we didn't know was a thing, especially in urban areas. And we see that habitat change is likely influencing long-legged flies' feeding behavior. So in total, you guys, I'm wrapping it up. My research shows that ecological filters do structure the community of predators that we found within the legacy city. Conservation planning should be placed on the location of the conservation site, not just what the conservation site is, the location of that site, how far vacant lots surrounded by other green space patches should be priority for transformation. So we should look at landscape variables before we go and implement conservation efforts in these cities. I would like to thank the Gardner Lab. We all really worked together, even though we all worked on different insects. We kind of all came together to kind of shed light on what was happening in Cleveland, Ohio. And I want to give shout out to the pictures you see at the top of my undergraduate worker, which couldn't have been done without her, which is why I stress undergraduate research so much at Prairie View. And I'm happy to be here, you guys. Any questions?
Thank you. Wow, what an outstanding research program. I think, Dr. Adu, we've been looking for an entomologist for some time, and you have one next door. No, seriously, I think this is it's a great research, and commend you for what you do.
Thank you.
But you're an Aggie, so basically this is home.
Yeah, it's home. Yeah.
So we'll open the floor for questions for Dr. Parker.
Thank you. I have 2 questions. The first question is, as an agriculturalist, how can you translate this urban and rural, just certain parts of rural? You did some community agriculture stuff like that?
Yes.
I mean, the garden-based studies, how you can translate your finding to insect pest management, just integrated insect pest management. That's the first question I have. Second question is just on your first result, you are comparing the control group, how you are certain on your conclusion said isolation of patch is a major factor contributing their abundance. How you are sure that the presence or absence of egg deposition at the beginning that creates the difference among patches?
Okay, so the first question was how I can apply it to just pest research in general. I think when you have to think about the key pests in the area, and then who are their predators, right? Who are their predators and what the research shows. So something that I think could be really key into Dolichopotidae in the urban setting is kind of looking at an agriculture setting and see what is the dietary niche breadth in the agriculture setting. That can kind of give you that realized dietary niche breadth on what they're capable of eating. And with that, that's how you can go ahead and implement some type of pest control. The same thing could be done for lady beetles. I didn't add the chapter. We did look at the diet of lady beetles in urban areas. I just didn't have enough of the individuals I didn't feel comfortable. It's another study, like another year or so. So I think that's also something that can be done in agriculture areas, or even just comparing one agriculture farm that is really successful to one that's maybe not, and seeing why that may be in terms of insects.
I think it's also urban agriculture is something that is being right now becoming a common. So I think whatever you're finding are
It can be applied.
related to urban agriculture. Yes. So there is a niche there. Urban agriculture is a reality. And I think if you look at it from carbon footprint, from energy footprints, urban agriculture is a viable, and it can be, if it cannot be viable on how many, 27,000 or 30,000 lots agriculture can
It
be
can
a
help.
source for food security for
can.
those food desert that you're describing in Cleveland, Ohio. So I think this is a great finding, in my opinion, that needs to be stressed. We have intervention here.
Thank you, Dr. Parker. Great subject indeed. I am curious, could you update us on the status of replacing grasslands in front of our houses with vegetables and urban gardens? Ever since I came to US, I'm really inspired by the grass, but
Yes.
I always think, why don't we put vegetable gardens? And any thoughts on that? Thank you.
Yes, I'm a real advocate for it. Fresh produce, fresh fruits and vegetables. It's really not hard. A lot of times, we had people managing the farms, of course, because we couldn't manage them ourselves. But being there and just seeing like the day-to-day interactions, or people coming to the community, they will literally set out a stand, and people could come to the community, get some things for free, some things for a pretty low cost. And I think just implementing that to anything, like if you live in a city, if you live in maybe if you have just extra land in your house, just just starting somewhere, find a crop that you know can grow easy, start there and kind of expand. And you can't eat it all, so share it with your neighbors, share it with the people in the community. And that's kind of addressing that food insecurity issue that we have all over the world.
But currently, Texas doesn't, HOAs don't allow the front yard to be replaced with either wildflowers or vegetable gardens. Do
It
they?
has to probably be a raised bed, I think, and then they really worry about how nice it looks. And that's like the real issue when you're trying to do agriculture on a large scale, even within a city. It doesn't always look pretty. So that's a good idea, just to see how that can be implemented in HOA. And we kind of just gave the city a plan. Like we just told them like, hey, this is what we want to do. This is why we think it's important. And they accepted it. We had to get their approval before even getting NSF approval for the grant. So just trying to work with local community leaders to see how you can address the issue, I think would be a start.
Well, I think that's, we have a question here.
Yeah, thank you. And just a comment, and then I'm going to ask you a question about your current role, if you don't mind sharing with us.
Okay.
So the comment is to students, you know, when you make a hypothesis and the answer is no, that's still an answer
Yeah.
and it's still data and you still get the degree. So I always tell students, don't worry, if the answer is no, it's not what you wanted to hear, but it's important, and it's important to still publish that data so that people know the answer is no, so somebody doesn't repeat it. So I like that in your presentation. So it's okay to say the answer was no. But then I'm going to ask you to tell us a little bit about your role here at Prairie View and what you do with undergraduates and undergraduate research, if you don't mind.
Okay. So my current role, one, I would like to agree with the first statement you made about no results is still results, because at the end of the day, we weren't doing it for the name of Ohio State and science. It was really a community project. So being able to give the community the results, we found like, hey, this may not have been as a good idea as we thought, or maybe we should try to look at it at another angle, which they thought was really helpful and really valued our truthfulness within that process. And then secondly, I'm currently the Student Research Development Officer in the Division of Research and Innovation. As of now, my job is basically to oversee the RISE program. So I supply faculty members with funding that can support undergraduate and graduate student researchers. I also co-host a conference for undergraduate students or student researchers in general once a year. That conference was about 2 weeks ago. Over 150 students participated in posters and oral presentations. So that's my role right now.
Well, I would like to recognize our graduate students who took time and participate. So can you raise your hand so we can see you? So our students here that are really, Kayla as well, you're a student, I think.
Just a quick comment, This was a great presentation, and as Dr. Ferris said, we have an entomology class, so we're thinking about it. Okay, so maybe we'll get you in the classroom soon, yeah.
I think there is more than just teaching. I think we definitely in research, we would need the service of, because we have, I mean, plant protection is, you cannot have agriculture production without
That's
having a strong plant protection program, and entomology is the encore of that. So
Yeah.
any other question or comments? We're gonna charge you for this one. eh?
My second question was not addressed, so I want to get a reflection on that. That's why. The egg deposition, you say, the habitat isolation, patch isolation is a major factor
Yes.
for the abundancy, but how you just control or how you make sure that there was no egg deposition difference between patch before your experiment? That was my question.
Oh, I get what you're saying. How can I test for that? Ah. that's a
But
good
I
one.
think it's the question is still valid in a way that even if you didn't, if you didn't, what you call it, if you didn't account for that or you didn't count it, they are already there because of the she's looking at the what you call it, the land use
Yes.
difference.
So basically, I
So-
get like an aerial view of a site, right? So the site, and then I use GIS to see what's around the site, and that's how I'm getting the calculations. I don't know.
So
until-
basically comparing the land
Looking.
use, the question is comparing the land use.
Yeah. I get it.
Or your approach
Mhm.
is permanent,
Mhm.
acceptable, acceptable. But my question is,
How to use it
I'm
in
seeing
agro.
the kind of control experiment. Whatever you manipulate the landscape, I'm looking, everything has to be the same at the beginning before the manipulation starts.
I get what you're
So
saying.
that I can compare with the.
So how do you find your control in the agriculture setting? That makes sense. That's a good one.
Sounds good. I like this discussion.
That's a good question.
Well, do you have any other question for our presenter? Yes, Dr. Memoli, please.
I just wanted to comment with regard to entomology that, yes, we are very interested in CARC research to introduce entomology research that is kind of centered around pollinators that are extremely important for food security. But as Dr. Parker presented, there are a lot of interconnectedness among the insects and the bugs there. They're actually, some of them are quite useful. There's a balance that exists. They developed sort of a symbiotic relationship between the fungi and the insect, and then insect feeds on something. So it's a fascinating world, and we are definitely interested in. Thank you.
Thank you, Dr. Memoli. Yes, Dean D'Souza.
No, let me take this in a slightly different direction. And since there are some students here, let me ask you folks, what are some barriers that you as students see for actually conducting research, if any? If you're interested in research, and hopefully Dr. Parker's presentation, and she's a great role model. Mississippi to Ohio State, did a PhD, and back here at Prairie View in an administrative and leadership capability. So from your perspective as students, what do you see as barriers to research here at Prairie View? Well, I didn't mean to put you on the spot, but here's an opportunity.
Yeah.
I don't know.
That's fine. Yeah, okay.
Reggie.
Well,
got
I think-
something.
Go ahead, Dean D'Souza, if you want to follow up. Barrier, but yeah, challenges.
Mhm. Oh, okay. I thought it was just money, because with money, we can make a difference. But equipment, no, there's other people here who can help with that.
Not TV, I think.
No, it takes a while.
Nothing we can do.
Well, I think we're very blessed with, in many respects, to that we do have resources. We do have great researchers and great students as well. So I think if there is barrier, it's ourselves. Okay, so if you don't have any other question, let's thank our presenter for an outstanding presentation. And we'll see you next week.